We truly look forward to a very informative and inspiring presentation this evening. And once again, thank you all for joining us, and I look forward to tonight's session.
Thank you, Prez. We'll now have the welcome and introduction of members and roll call of visiting clubs, and this will be done by VP Uncle Andrew Burton of the Kiwanis Club of Kingston.
Hello, good evening, everyone. I'd like to extend, this is a joint meeting, so if I can take the privilege on behalf of both Presidents Zuri and President Giovanna to welcome you all to our meeting and our exciting topic, as we've heard in their opening remarks and as we've seen on the flyer, No Cell Left Behind, is if our guest speaker is on the call. I welcome her, Dr. Badal, representing one of the best, if I can pull that bias, one of the best faculties at the University of the West Indies as one of our incoming deputy deans. So I look forward to what she has to say. And if there are any guests who are here, especially to hear from her, even from outside of Kiwanis, we welcome you. So I open the floor to anyone from Kiwanis clubs outside of Jamaica. Please unmute your mics. Share with us, tell us where you're signing in from. Okay, any clubs from Division 25? Anybody from Kiwanis clubs from Division 25?
Good evening, everyone.
Good evening. Hi, Prez.
All right, so this is President Giovanni from the Kiwanis Club of Coastal Trelawney. And with me so far, well, there are 5 of us on. I have Chairman Anne-Marie, DP Dorrit, DP Tonian, and also Chairman Rochelle. Thanks for the invite and looking forward to a very informative meeting and also other members from our club joining.
Excellent. Welcome, President Giovanni, and your delegation. Any other clubs from Division 25? If not, any clubs from Division 24? Kiwanis clubs from Division 24. Okay, Kiwanis clubs from Division 23 East.
Good evening, everyone.
Hello, good evening. I see.
Good evening from Cyber Connect Jamaica. I wish you guys to have a fabulous meeting. No Cell Left Behind sounds like a very interesting topic. Right now, I am single, but I'm sure more will join for this interesting session today. Thank you very much for the invitation.
All right, singly joining us thus far, but hope others will join. Thank you so much for joining.
Good evening.
Good evening.
This is President Curlin from the Kiwanis Club of Trafalgar. I'm bringing greetings on behalf of my club. We have 4 members on plus 1 guest, and I'm really looking for a very informative meeting. Thanks for the invite.
Welcome, President Curlin, and thank you for bringing your delegation, including a guest. We always welcome that. Thank you so much for joining us.
Good evening,
Good evening.
VP Andrew.
Good evening.
This is DS Bridget from the Kiwanis Club of New Kingston, and I bring warm greetings from our President Veronica and all the members of New Kingston. Joining me this evening, I have 5 other members, including DLG Gertlin, DP Val, and DS Lindo. I have Marlene Owen and Claudette Walker as well. We are looking forward to a very informative meeting. Thank you so much for the invitation.
Excellent. Thank you, DS Bridget, and your delegation.
Good evening.
Good
My name is
evening.
Erica Watson. I am the secretary of the Kiwanis Club of Dynamic Leaders, Jamaica
Excellent.
Online, and we have 4 members on. Looking forward for an informative lesson.
Okay, dynamic leaders, welcome. Thank you for joining us. Okay, is that somebody else from Division 23 East? No? Okay. Any clubs from Division 23 West?
Good evening,
Good evening.
fellow Kiwanis. Hi, this is President Wayne joining from the Kiwanis Club of Portmore, bringing greetings on behalf of the team. With me currently are President-designate Henry, Director-designate Noel, and DP Donnell. Yes, so thank you very much for this interesting invitation. Looking forward to this meeting. Thank you.
All right, welcome. Thank you for joining us, as usual. Any other clubs from Division 23 West? Okay, the 23 Central Posse. Please unmute and declare where you're from and what your delegation size is, etc.
Good evening, VP Andrew.
Evening, Pres.
You hearing me?
Yes, yeah, man, we're hearing you.
This is President Demar here from the Kiwanis Club of Constant Springs. We are currently 18 numbers, and we are expecting a few others to join. We want to thank you for the invitation, and we are looking for a very informative and interesting meeting this evening.
All right, thank you, President Damar, and the group from Kiwanis Club of Constant Spring. Welcome. Other clubs from 23 Central?
Hello, good evening, VP. How are you?
Hello, I'm well, thank you. How are you doing?
I am wonderful.
Good.
Great. So, as you know, I'm DP, you better say, from the
Yes.
Kiwanis Club of 23 Central Surrey Online. And at this time, I am only seeing well, not true. I'm going to borrow DLG Desmond.
Always. We share him.
Yes, so we have a delegation of 2.
Okay.
And I'm looking forward to this presentation. You know, I see it's a kind of play on the military, no soldier left behind.
Yes.
So I'm all fired up for this topic. So looking forward to it. Have a great meeting. Thanks.
All right. Excellent. Thank you, DP. Welcome.
Mhm.
Any other clubs from 23 Central?
23. Good night.
Good night. Evening, welcome.
I see 2 other members online.
Okay.
Yes, and
Jella.
I expect to I know this will be a great meeting, and thanks for the invite.
Okay. Thank you for joining us. Any other clubs from 23 Central? Okay. Any of our service leadership programs, any of our SLP members?
No, so I think I see Ashira, PP Ashira from Nation Builders. Anyway.
Okay, excellent.
Yes.
Nation Builders in the building. Welcome, PP. Okay. Do we have any of our SLP members? Any guests of Kiwanians?
VP Andrew,
Yes. How you did, DLG?
you may want to welcome back home
Okay.
past Treasure Orville Lewis, who we haven't seen for a long time online.
Excellent.
Wow.
Welcome home. Welcome home, Pastor Treasurer. Good to see you in the meeting.
Thank you. Thank you. Thank you, VP.
We have a Dr. Berry, an intern from Spanish Town Hospital, who's joined us, was invited by a Kiwanian, and has strong interest in the field being presented. Welcome, Dr. Berry, to the meeting. Glad to have you, and you're always welcome to come back, to return.
Thank you.
I have a guest. Oh,
Yes, okay. Yes. DPC,
yes,
please.
I have a guest. She's from Trinidad, June.
Yes.
I saw her name, so welcome, June. Glad you could make it.
Excellent.
Back to you.
All right, thank you. And always special welcome to one of our prospects who has been so present, in person and virtually, Prospect Stephen. Welcome to the meeting. Any other guests? Any guests of Kiwanians? Prospective members, all, we welcome you.
Uncle Andrew, if
Hello,
you
DLG.
will allow me, I know I invited Dr. Karam Smith, and he's
Okay.
on.
Okay, welcome, Dr. Smith.
He don't want to say hello, but he is on.
Okay. Welcome, Karim.
There he's greeting you.
Good night, good night. I'm here, but the background is very loud. I'm on call at Spanish Town Hospital.
Oh, got you.
Hey, Doc.
Okay.
What's up?
All right. Hey,
Uncle Karim.
good to have you in the meeting, Karim. All right, and an internal welcome to all members of our host clubs this evening, members of the Club of Kingston, members of the Club of Barbican, my new friends from our recent trip to Murray Atkins, and we look forward to the continued partnership through both meetings and service. I return the mic to President Zuri to continue the program. Thank you.
Thank you, VP Uncle Andrew. I have no apologies for absence. I do, however, have one for lateness, and that's from DP Newton Randall, who did indicate that he would arrive late to the meeting. I'll ask President Giovanna if she has any apologies.
Press, we have apologies from DP Kerry Solness. She is not able to join tonight's meeting. That is all from my side.
All right, thank you, Prez. DLG, Willie, did anyone send any apologies to you?
No, I didn't receive any apologies.
All right, thank you, DLG. There being no further apology for absence, we move to recognition of birthdays and anniversaries. I'll invite P. Arsenio to lead us in this section.
Good evening again, everyone. So this is the segment of our meeting where we just like to take a moment to highlight those who have special days coming up. So that will be between April 7 and April 13. We have an anniversary on the 9th, and that is for Kiwanian Gregory Gibson and his wife Donna. Happy anniversary to you both when it comes. Then on the 13th, we'll have a birthday, and that is for Kiwanian Claribert Scott. Happy birthday to you when it comes. If there's anyone else on the call, be it from Barbican or any of our visiting clubs or guests that have a special day coming up this week, feel free to open your mics and share with us.
I think
Hi,
President Giovanna is going to be.
Yes, good evening again. So from the Kiwanis Club of Barbican side, we have 3 birthdays in April. We have IPP Kevonia Tomlinson, we have DP Kerry Solness, and we have DP Chantal Dorma, whose birthday is this month. And that's it for my side.
Thank you, Prez. PER Senior did give the invitation to anyone on the call who is celebrating a birthday or special occasion to, if you wish to share with us, we give you the opportunity to do so now.
Good evening again, fellow Kiwanians. Good evening, President Giovanna and President Zuri.
Good evening.
President Demar here from the Kiwanis Club of Constant Spring again, and I will be celebrating my birthday on Saturday, April 11th.
All right, Prez, we'll celebrate with you. All right,
Thank you
let
very
us
much.
know where the party will be and we will
Not
turn
a
up.
problem. The whole week of celebration.
Is there anyone else on the call who will be celebrating a special day, a special occasion, a special achievement? All right. Well, we wish all the persons that are celebrating all the very best, and for those celebrating birthdays and anniversaries, we wish for you many, many happy returns. And at this time, we will have Kiwanis Education done by DLG Desmond Bachelor.
Thank you, President Zuri. Good evening, fellow Kiwanians, SLP members and guests. Of course, just to say, first of all, BP, I hope that Clarbert Scott could hear us, just to tell you that he passed on about 4 years ago, 3 or 4 years ago. Number two, DP, you already see our President Maxine is with us. But the topic this evening is annual general meeting. But can anyone quickly tell me the purpose of the annual general meeting? All right, well, I'll answer. It's to elect officers and directors and hear reports from the club president, the secretary, and the treasurer. And I'll just stress those, as ever so often we don't have those reports. The club president, the secretary, and the treasurer. We hold the annual club meeting during a regular club meeting between January 1 and May 15th. We must give 30 days notice of the AGM, the annual general meeting. 5 weeks in advance, the president appoints a nominating committee to make nominations and prepare a ballot to elect officers and directors. 2 weeks in advance, the nominating committee submits a list of nominees. For officers, there should be no more than 2 nominees for each office. And for directors, that means no more than the number of directors. To fill the position, plus 3 candidates. The president-elect is the sole candidate for president nominated by the committee 1 week in advance.
1 week in advance.
Oh,
Yeah, 1 week in
you're
advance.
on.
The nominations from the floor.
Okay, I just didn't see you on the meeting. I just tried to, I didn't know what name you had on. Okay.
Yes. Nominations from the floor may be made for any position to be filled. At the election meeting, volunteers will be appointed to prepare the ballot, count votes, and certify results. And we want to say that additional nomination from the floor may be made at the meeting. And I'll just go on a little bit further to say that only active members in good standing may vote. It's 1/3 of the members present and voting will form the quorum. So those who are voting are those in good standing. And 1/3 of that number would form the quorum. I'll stop there, President Zuri. There could be more, but that's it for now. Thank you very much. Over to you, President.
Thank you. Thank you, DLG Desmond. The fact that he stopped there means that he's giving you all the opportunity to continue your reading outside of this meeting so you can go and find out more about the annual general meeting and how it is that your club should be conducting this meeting. Like I like to share, say try to learn as much as you can about the organization that you serve. So try to learn if something new, if not daily, let's try for weekly. But try to see how best you can increase your knowledge about the organization that you are a part of and that you serve so well. So we are at the main event, no cell left behind. And I'm going to be inviting Dr. Martino Dyer. Welcome to you, sir. It's been a while since we have seen you as well. It's good to have you home. And he will be doing the introduction of this evening's guest speaker.
So I get the pleasure to introduce our guest speaker, Dr. Badal, who was actually one of my tutors at UA when I was doing my first degree in biochemistry. And it's so good to see her doing big things in an era which we definitely need more insight in. So Dr. Simone Badal is a senior lecturer and the incoming Deputy Dean of Research in the Faculty of Medical Sciences and cancer researcher at University Hospital of the West Indies, Mona, where she leads the Anti-Cancer Research Jamaica Laboratory, which is the only advanced cancer cell biology facility in the Caribbean. She's a 2025 laureate of the Anthony N. Sabga Caribbean Award for Excellence in Science and Technology, which is widely regarded as the Caribbean's equivalent of the Nobel Prize. Her work centers on a deceptively simple but urgent question. Why are cancer that disproportionately kill people of African ancestry studied almost entirely in cells derived from people who look nothing like them? Her answer has been to build what did not exist, which is a Caribbean-derived cancer cell line now used by researchers around the world. It is supported by over 1 million in competitive international funding from institutes such as NIH and the Chan Zuckerberg Initiative. That conviction runs through everything that she creates, from her Springer Nature cell culture textbooks, which are reshaping how next generation of scientists is trained, to her memoir, No Cell Left Behind, which is a deeply personal account of her resilience, faith, and refusal to be overlooked that have defined both her science and her story. Dr. Badal, welcome. President Zuri, can you hear me?
I can.
I don't know why my thing sounds a little low, but if you can hear me, that's fine. There's just a slight little mix-up. I need to give Dr. Badal another minute or two at the most to come on. All right?
Okay.
Because I was searching for her name, but just give her another minute or two. In the meantime, maybe I could just welcome another guest that I had, and I didn't see her name, so I don't want to have mentioned one and not the other. That's Janet McPherson. I just see your name. So if there's anything else that you want to use to just fill another 2 minutes, she'll be on momentarily, please.
Okay, all right, thank you. I saw DP Marcel on earlier. So we could probably take the thought of the week now while we wait.
Hi, or good evening, all. Yes, I'm happy to do that. So we can do salt for the meeting, salt for the evening, and salt for the week. Stop worrying about what can go wrong and get excited about what can go right. And since we expect a lot of things to go right with the presentation this evening, let's get excited. So stop worrying about what can go wrong and get excited about what can go right. Over to you, President.
Thank you. So we won't focus on what can go wrong or what has gone wrong, but we will more focus on what will be right with the presentation. Dale G. Willie, did notice and announcements, or if Secretary Dion is on from Barbican and you wish to go first, ladies before gentlemen. Well, either Secretary on. All right. So I guess I'll just catch up and ask how has your week been going? Hoping that you've been having a good week so far. And here comes DLG Willie to give us notices and announcements and tell us about interclubbing opportunities.
Thank you, President Zuri.
You're welcome.
As to notices, reminder that the directors meeting, it was mentioned earlier by the President, is on Monday, coming Monday, 7 p.m. at the Secretariat. There will, more importantly, this week is celebrated as Key Club Jamaica District Convention at Manille Teachers College from Thursday the 9th to April the 11th. Kiwanis, please note that if you're going down, you should have your background check. If you don't, there will be an entrance fee of $5,000 if you want to participate in the activities and lunch provided by the district, Jamaica Key Club District. Upcoming fundraising events, Barbie Pride, Friday, May the 29th at 11 Fairway Avenue at 10 a.m. and on Saturday, July 18th at 8 p.m., we have the benefit performance of the National Dance Theatre Company. We are fortunate to get the second night as the subsequent night after their opening night. So we look forward to the support of Kiwanians at both Barbie Pride and the National Dance Theatre benefit performance. Respecting the clubbing opportunities, as usual, I will circulate the schedule of upcoming interclub opportunities in the morning. Thank you. Back over to you, President.
Okay, President, she's on.
Okay, all right, thank you. Thank you, DLG. So we will go to our guest speaker, and then after our guest speaker, then we will have the notices from Barbican.
Hi. Thank you for having me, and I apologize for being late. I got the dates mixed up, but I'm happy that I'm still here to be with you. So I originally told Stuart that I wasn't going to give a PowerPoint presentation, and I still am not going to. I just want to sit and talk to you. And I hope that is okay, but if you would like some slides, I have those on standby I can share with you. So once again, I am Simone Badal. I'm a senior lecturer at the University of the West Indies, and I am a cancer researcher, and I've been at this for some time. And my journey started during my PhD days when I wanted to investigate two areas. We had, at the time, a lot of Jamaican natural products that were not tested. So we wanted to see, one, if these products could prevent cancer, and two, if they could treat cancer. So my journey began then, some time ago. If these products could treat cancer, how would they be able to do that? There are specific enzymes in the body that we call drug metabolizing enzymes, and the scientific term cytochrome P450 enzymes. And these enzymes tend to convert certain entities that we inhale, entities that we eat, we drink, into other entities that are cancer causing. So for that aspect of my project, I wanted to see if our natural products could inhibit the activities of these enzymes. And if so, then perhaps they could prevent the cancer-causing entities to be formed in the body. And we found quite a couple of them that could do that. The second part of my project was looking at can our natural products treat cancer? And if they can, to what extent? To do this, we employed the first step in the drug development pipeline. So in the drug development pipeline specific for cancer, we tend to look at the cells. So we call them cell lines. These are cells that we take from the host, and for this specific area, it would be human beings. We grow them outside of the body indefinitely, and we expose them to different agents. And we can answer so many questions doing this. So can our products kill cancer cells? And if so, can they not harm normal cells? So that was the aspect of my project that I was very interested in doing. So at the time, we didn't have the facility at the University of West Indies. So, I traveled abroad to the States at the Clemson University, and I looked at different cancer cells. I looked at colon, I looked at breast, I looked at liver, and I also looked at normal cells. And I had over 20 natural compounds and some synthetic compounds as well. We exposed them to these cancer cells, and we also exposed the cancer cells to anti-cancer drugs because we wanted to see if our products could kill these cancer cells, to what extent would they kill the cells? Would it be similar to known drugs on the market? Would it be better? Would it be worse? Would they be safer? And at the end of that portion of my PhD, we found that several of our products were more effective in killing the cancer cells than known anti-cancer drugs. And what was more interesting for me is that some of our products did not harm normal cells compared to the known anti-cancer drugs that were also killing the normal cells. So of course I was excited about that. And I was able to wrap up the degree in a relatively reasonable time frame. We published several manuscripts, won several awards. And because of the significance of the data, we were able to generate funding to go on. So I'm like, all right, this is awesome. We've checked off the first stage. What is the second stage in the drug development pipeline? The second stage is looking at these entities in animal models. So we can check off that they're effective in the cells. The second stage is in animals, and the third and final stage is in humans, clinical trials. So my intention was to take this research all the way. So of course, I was interested in progressing to the second stage. We didn't have the facility, and we still don't. And to employ that stage, it takes quite a bit of money. So I sat down, I started writing grant proposals, started trying to get funding. And I submitted applications to the National Health Fund. But while I sat with the data on a high, just finished a wonderful degree, published several pretty good papers, and we're getting the attention from this research. I took a step back and I got a different perspective on the work that I had done. So yes, we wanted to look at the effectiveness of these compounds in terms of treating and preventing cancer. But what type of cells was I using? And I realized all the cell lines I used in my PhD, cell lines that I purchased, or we purchased from a main supplier called American Type Culture Collection in America, one of the global supply of cell lines. all the cell lines I used were cell lines from Caucasians. And so I stepped back. I had graduated already, degree is finished, but it got me thinking, if I used all these or tested these compounds in cells from white men, white women, would they be equally effective, less effective, or more effective if I tested them in cells from our own people? And so I started looking for cells generally from Blacks, and I could hardly find any. I started looking for cell lines from Caribbean people, and there were no cell lines. So, this was a very critical juncture for me. I could have progressed to the next stage, and I'm certain if I did, I would be much further along in terms of the drug development pipeline, and personally could be better. But I wanted the work that we were doing to be first and foremost effective in our people. If we're exploiting or trying to find the anti-cancer or chemopreventive efficacy of the products that reside here, then those products should firstly be effective in our people. So I said, okay, before I pause and before I go to the second stage, perhaps we need to revisit the first stage, and perhaps we need to redo these experiments, testing these models in our own population. And so we needed to develop our own cell lines. And of course, I have no idea what developing a cell line means. I've just tested the compounds in the cell lines. So I would have connected with different persons who had explored that avenue, who were experts in that avenue. As a matter of fact, one of the persons that I contacted to work with me along this, there was a publication where he announced that he had created a prostate line in America, an African prostate cell line. So I reached out to him and I said, hey, I'm doing this work and I'd like to develop cell lines from our Jamaican population. Would you be willing to work with me on this project? And if I had pulled up his email and read it to you, you would be in awe. He said, no, I don't recommend that you do this work. This is very high risk work. I recommend that you do something else. And later on, it was found that the cell line, the prostate line that he had developed wasn't actually prostate, it was kidney. And it wasn't from a Black man, it was actually from a white person. So that was pulled from the market. And there's someone else that I reached out to, and he said, Simone, I have tried developing cell lines. This is someone with a fully funded lab in the States, in Miami. I've tried it and it's never worked for me. If I were you, I wouldn't. If you want to, fine, go ahead, but I can't help because I have had no success with this. We had persons come to the university, I presented and I said, look, we have all these potential compounds with all these wonderful properties. We need to test them on our own models. I need help. And persons would be like, Simone, I don't recommend that you do that. So I sat down and I weighed all the pros and the cons, and I decided, after prayer, knowing that I believe this is where the Lord was leading me, that it was not enough to say that the research was risky to not do it, because the end would necessitate the journey to getting there. So of course, what do we do as scientists? We get papers. There are persons who develop cell lines. It's not a novel thing, and it's a course that I teach at UA. And as I evolved and acquired more information along the journey, I realized that cell line development is actually very high risk. The success rate is under 10%. In the literature, we have documentations from in the 1800s where scientists attempted to develop human cell lines. And it wasn't until 1950 that the first human cell line was developed. And maybe some of you have heard of her name, Henrietta Lacks. A movie was developed on her, starred by Oprah Winfrey. And her story was documented by an author called Rebecca Skloot. So she documented the journey of Henrietta Lacks and how her cells opened the doors for the revolution in biomedical research, and especially in cancer research. So it's from the 1800s to 1951, that's a very long time. And here it is, it's 2026. At the time I was doing this, it was like 2015, 2016. A lot of advancements would have occurred, but still the number of cell lines that we have access to as researchers, it's limited when you look at the variety of tumors that exist. So I was in for a long road ahead, but determined to get this done, because as I researched more, recognized more, I realized that the drugs that have been developed are not as effective in our people as they are in white people. The data is showing that. And so this research was and remains critical to treating cancers in Black people, in Caribbean people. So we got funding. We were successful in getting funding from the National Health Fund, and I used it to set up the Anti-Cancer Research Jamaica Lab, so a cell culture lab at the University of the West Indies. My first student, PhD student, Henkel Valentine, he took the journey with me. And it was, for us, as we were going through it, a long journey. There are times, tears, times when we thought we were going to get this thing done, and it just did not work. And in 2019, December 2019, Henkel wrote me and he said, Dr. Badal, I think I have some cells. And at this point, I was cautious because I had sent Henkel to the Fox Chase Cancer Center earlier to get it done. And we thought we had done it, but when they came back with the authentication report, it indicated that we hadn't done it. So, I was very cautious and I said, okay, continue to watch it. And in early 2020, when we were on the brink of going into the COVID era, we got the wonderful news that we were successful in developing the first cell line from the Caribbean, a prostate cell line, which is now published, and it is now licensed with a cell line supplier in Canada, and ATCC, the global cell line supplier, has been asking for our cell line to have it introduced in their bank, and that's something that we are working on doing. And since then, thank you very much. I see the claps. Since then, we have expanded from that one prostate line. We developed 3 breast lines, and I was actually just working on that manuscript because the publisher came back with edits. So hopefully that manuscript should be published soon, and we can also deposit that in the cell line banks. And we have developed additional prostate lines, but those have not been characterized yet. And the goal is to use these models. We know that AI is here, it's going nowhere, so we better be comfortable with it. And we know that AI is going to take over. I mean, Elon tweeted some time ago that if you are looking at becoming a doctor, he advised not to do it because soon their robots will be able to do that. AI is going to be very critical in precision medicine, in personalized therapies. And we need these AIs to be trained in a holistic manner. And so we need ourselves so that we can train the AI with the cancers from our people, so that when it's there to diagnose and to develop treatments, those treatments are indeed reflective of the tumors that they're trained on from our people, and not treatments that are just there representing one set of persons. Already I'm seeing in a paper that we just published last year, where we screened our prostate or novel prostate line against several anti-cancer drugs, and we compared the effects with other prostate lines from white men and a prostate line, I believe, from another African-American man. And we're seeing differential responses. There's one particular drug that was way more effective in our prostate line. And to that end, I'm developing a clinical trial grant application to conduct a clinical trial, because if it is that the therapy that or protocol that's mandated by the Ministry of Health is using a drug or a series of drugs that's not as effective, then perhaps we need to change that. So we need the data to inform us of that. So Ursa Lines, they're coming along. They're doing what they're supposed to do, which is to inform us as to the best treatments. And the long goal is to use them to develop or to advance or transition our products into more effective therapies, into safer therapies for our men with cancers, for our women with cancers. And so I published this book in 2023, which is the title of the talk today, No Cell Left Behind. And it is on Amazon if you want to. I just briefly went through the journey from starting to where we are now. And as much as I would say we've done a lot, there is still so much more work to be done. And I'm happy that I'm taking this journey alongside many persons who are working with me. My students, I mentioned Henkel, and there is Shanique Clark, who developed the breast lines. There are so many of them. There's Isabella Allen, who's gone back to the States, and I have so many more in my lab. Christoph, Frank, Chelsea, James, and others. And I have a series of clinicians working with me. I have urologists who supply me with tissues. I have radiologists who give me breast biopsies. I have oncologists. I have epidemiologists, statisticians working with me both locally and abroad, because this is not a one man or one woman show. This is a team effort. And together with this singular goal of ensuring that our people are represented on the research stage when it comes on to understanding cancers and when it comes on to developing treatments for cancers in our people, that is our mission. And we don't take that mission lightly. We live, we breathe in that mission. And I'm going to wrap up. I hope that wasn't too short or too long, and welcome any questions you may have.
Thank you very much, Simone. You used the word in your presentation, and I just wanted to just get that out of the way. When you said that you had developed some cell line, but it had not yet been characterized, what does that mean, the word characterized in this context?
All right, so someone asked, what is a cell line? So let me define that first, and then I will say what characterizing means. So a cell line is pretty much cells that we take from the body and grow them outside of the body indefinitely. So they are immortal. So for example, the HeLa cells that I described that were developed in 1950, 1951, those cells still exist and we can still purchase them from ATCC for use. So if once you're able to grow those cells indefinitely, you have a cell line. What does it mean to characterize that cell line? So I go in the lab and I say I'm going to be developing a prostate cell line. So I get the prostate tissue from the urologist. I take it to the lab, I process it, I do all the works, and it took us 5 years to develop the cell line, by the way. And I come out and I say, I have these cells, okay, on what merit? You know, I have to then prove that one, these are novel. So we send them to we can send them to like Carigen that's on campus that does a series of tests to compare or to ensure that our markers in the cell lines are indeed novel. So they have like a standard registry that shows, you call them short tandem repeats for all the existing lines, and they compare our line to it. So that's the first form of characterizing to ensure that this is indeed novel. The second is this from a male? Is this from a female? So you test if it has XX chromosome, XY chromosomes, and you go through additional things. How can I say that this is prostate? So you have to test if there are prostate-specific biomarkers or if there are breast-specific biomarkers. So characterizing simply means that I am running a series of tests to prove to you that what I'm saying is prostate cancer is indeed prostate cancer. So if you are able, our manuscript is online. I am also happy to share that with you. You will see all the tests that we had to go through. So after spending 5 years to develop the line, that was just a small component. We had to spend a couple more years to run all these tests. And we did those tests with our collaborators overseas, primarily Fox Chase Cancer Center in Philadelphia. And I'm happy to report that through funding that we've acquired from the Chase Fund, we are getting some high-tech equipment and we'll be able to do most of those tests here at home. And that's also been one of my dreams, to reduce our dependency or reliance on overseas. Why can't we do everything here, you know? And we're making all those wonderful steps to be self-reliant, self-sustained.
Okay. The audience is welcome to put up your hand. And I see one now. Paul, is that your hand or was it a clap?
It was a clap, but Miguel Baker has his hand up.
Okay, go ahead, Miguel.
All right, good evening. Can you hear me?
Yes, we're hearing you.
Okay. So I'm a hematology and oncology resident. So I'm actually just in my second year, and I always say I want to do a fellowship, and I was considering doing it in stem cell research and stuff like that. But since I technically would be the clinician in this case, when I was doing the research and I was looking up how to get stem cell transplant here in Jamaica, I noticed a lot of roadblocks in terms of us not having the resources in terms of the blood supply that we need, the isolated area, and all of that. But your research sounds very promising in regards to that. So I was wondering how far are you in terms of your own stem cell therapy being ready to be used in our generation of people?
Okay, so these are two different things. Stem cells or stem cell therapy is different from cell line. Cell line is simply a tool that we have developed and that is on the market and will be on the market for other researchers to use. And we use this tool to understand cancers, and we use this tool as a vehicle to developing anti-cancer drugs. Stem cell therapy is something completely different, but I'm certain that to do stem cell therapy, you would also, I believe, need a cell culture lab. And that's a lab that we currently have for our purpose. And also, I am in the process of developing another lab so that other persons in the faculty who are embarking or who need cell culture facilities can use that. So we have that going. In terms of our cell lines and how far along, as I said, developing the lines is in itself was a momentous task, but we still are far along in terms of where we need to go to develop drugs, right? So we tested our natural products when I did my PhD on a series of cell lines, and those cell lines were primarily Caucasian cell lines. We now need to go back and test those same products on cell lines from our people and do a comparative analysis. And we haven't yet done that. The stage that we're at now is testing current anti-cancer drugs to verify the differential impact that current anti-cancer drugs are having on our cells relative to other cells. So this is like proof of concept, if you want to say that. So we still have a long way to go, but we are uncovering the answers and we're seeing differential impacts. And so far, if we get the funding, so to run clinical trials, and that's not something that we do every day here at UWI or even in the Caribbean, it's still in the early days, it's something that takes a lot of money. So I've been putting grant applications together that will cost like 2, 3, 4 million pounds to get done. And that data will take like a couple of years to get. Once we get that data, then we'll be able to collaborate with the Ministry of Health to say, hey, this protocol that you have going for prostate treatment, we need to amend that because we're seeing another drug that's more effective as a first-line therapy. So perhaps you could use abiraterone instead of paclitaxel. That's where we're at now. And of course, we can do things simultaneously, but we need more hands and more funding. So we still have a long way to go, but we still have also come a long way. Hopefully I've answered your question.
Okay.
Yes, Doc. Thank you for the clarification.
Okay, I can't see my full screen. So if anybody has their hand up, President, you can tell me if you have seen it because I may not.
Well, I saw a hand up from, I think it was DP Valerie.
Okay. Let me ask DP Valerie if she'd like to come in now.
Well, Doc, this is very interesting, but I heard you say that the development of the drugs will take a little time from your research. And I was just asking right now, because I have so many friends and family who have passed on from cancer. And it seems to me that chemo, they've all said that chemo, many have had to stop because it destroys more than it helps. And I'm just wondering, what impact does your research have on the administering of chemo drugs, which seems to be really, as far as I'm concerned, it helps, but it does more damage than good.
That's a
How
very
has
good
your
question.
research helped?
Right. You know, research has shown that persons on chemo who then get your cancer-free report get a recurrence of cancer. It could be as the same cancer or different cancer. And research is showing that typically sometimes it's the chemotherapy drug that predisposes the patients to future cancers or additional cancers, which eventually takes them out. And this journey for me is personal because while it started out as a professional career, something that I needed to do and do well, it became personal because I was a part of a female Bible study group. And one of us developed cancer. Her name is Annie. And I remember we were at one of our usual Bible study meetings and she shared the news with us. And of course, it was devastating. The silence was deafening when she said, when she gave us the news. And we took the journey with her to the point that she was cancer-free. And then 1 to 2 years later, she was diagnosed with liver cancer. And she's no longer with us. And so this became personal for me. So what we realize is chemotherapy drugs, while they have been effective, they not only target the cancer cells, they also target your normal cells, specifically rapidly growing cells like your liver, your gut cells, your hair follicles, which is why you tend to lose hair. And it also compromises your immune system. And if your immune system is compromised, your fight back is reduced. With our model, we will be able to identify therapies that are effective in attacking cancers and simultaneously not attacking normal cells. In my talk earlier, I had mentioned that we saw some compounds that were effective in killing the cancer cells. There's one compound called scopolamine. It's a coumarin, and we had isolated it from Castella macrophylla, the plant. And that compound was effective in killing the colon cancer cells, comparable to the anti-cancer drug that we used, fluorouracil. And fluorouracil, when we tested fluorouracil on the normal colon cells, fluorouracil actually was cytotoxic, while scopolamine didn't impact the normal cells. So having our cell lines, we will be able to do this type of testing and tweaking. And the cancer treatment is evolving. It's evolving from one cancer drug or two cancer drug being given to a patient over the duration of the cancer. What we are realizing is that cancers are designed and built to survive. So while a doctor diagnoses you with ER negative, PR negative, HR, HER negative cancer, breast cancer today, that breast cancer can evolve into a triple negative breast cancer a week down the line, a month from the line. And so if you don't have that mindset where you have the constant testing and that cocktail of drug, not necessarily one drug, but a cocktail that is effective in attacking the cells, while at the same time boosting your immune system and simultaneously not harming your normal cells. Until we get to that stage, we're going to be stuck with 1-2 drug treated during the course of the cancer, and eventually you just pass from it. It could be 1 year, 2 years, 3 years. So our lines will ensure that the treatments are first and foremost effective on us, or population, using our genetics. And secondly, we can train the AI models with these lines so that these models can also effectively come up with a cocktail of drugs. And over time, during the course of your treatment, you're constantly testing that tumor. Is it the same tumor or has it evolved? So yeah, we're making steps there. We're making steps.
Thank you very much, Doc. That was clear, you know?
Doc, some years ago, we had as a guest speaker, Dr. Williams, I think that's his name, who had done a lot of research, I think, in the guinea hen weed. Are you familiar with that at all? Since you said you had looked at some local.
I am familiar. I did not do that research myself. I believe Dr. Lawrence Williams, is that who you're referring to? Right. I do believe he identified some compounds that were quite effective. I'm not certain what cell lines he used. I'm assuming he used cell lines that are standardly used, perhaps the MCF7, the HT29, and these are all from white people. I can always go back and corroborate, but I do remember his he had identified some very promising compounds from that plant. As to where his research is now, I'm not sure. The last time I remember, there was a pharmaceutical company that was interested in taking that over. I'm not certain where it is now, but I do recall, and it would be good if we could run those tests on our cells to see what the effect is on our cells, because it could be that they're not as effective, and it could be that they're more effective, you know. So that's something that we need to consider.
Okay.
Right.
Any more questions?
Yes, we have in the chat, so we have one from Oswald Newell, and he's asking Doc if a cell line can be developed from blood.
Yes. So you have suspension cell lines and you have adherent cell lines. So the adherent cell lines, typically the epithelial cell lines, are the ones that I am specialized in. I have never used blood cell lines, but you do have them.
And
Thank you,
DP
Doc.
Marcel has his hand up.
Okay,
Just
go ahead,
before
DP.
DP Marcel, let's just deal with the other one in the chat.
Okay.
What do you feed the cells to keep them alive? And what happens if the cells get contaminated?
If the cells get contaminated, it depends on the degree of contamination. If it's something mild, then we can use antibiotics, and we typically use pen strip in the lab. That eradicates them, and if that doesn't work, you simply have to throw the cells out and you have to start with a new batch of cells. We feed the cells with what we call media, and these are specialized liquid forms or stocks that we purchase from different suppliers. And each type of cell, you have a media that is specific for each type of cell. So like you have KSFM that we have used with our prostate lines, you have other types of media, PBM, you have natural, you have defined media. That's a whole lot of different science. But we purchase these, and important is that our technique is spot on. The lab has to be fully sterile. My students know that they can't play around with sterility, and especially if you have multiple persons using the lab at the same time, because contamination is indeed one of the biggest hurdles that scientists have had to cross to successfully develop cell lines. And the moment you have a contamination issue, that can erode years of research, and if you're unable to get that under control, you're screwed.
Back to zero. Yeah, back to zero.
All right, thank you, Doc. All right, so we move on to DP Marcel.
Oh,
Marcel, we're hearing
yes.
you.
Professor, yes, yeah. Sorry. Professor, this is fascinating. Absolutely fascinating. DP, Stuart kind of alluded to it. In hearing you, what came to my mind, my concern, here has always been the preventative, and you mentioned the immune system and all that, right? Two things. In carrying out your work, your wonderful work and your research, is a component of that to deal with the prevention, or can that be an offshoot? And if so, I know persons who have, you mentioned guinea hen weed. I read extensively and asking you know about soursop. I've seen and heard on persons who swear by it, who have knocked out and get cancer free. Using soursop. What's your view on that? It's particularly one, the preventative, the natural stuff like guinea hen weed and soursop, and in particular soursop.
I have heard that too. And what I heard was that the scientists who were working on soursop, they were trying to extract and synthesize the ingredients from soursop, so that of course they could transition that to drugs or pharmaceutical compounds, and they couldn't do it. And because they couldn't do it, I was told, I'm not certain if this is true, that they had locked the research down. One person who leaked it to say, hey, soursop is amazing. But guess what? It's not just soursop. There are so many natural products that are amazing. But you know what one of the problems is? One of the problems is that we're consuming these natural products that do not have the metabolites that we need them to have. What do I mean by this? We are, or plants, the God in his great divine wisdom created these plants to also develop quote unquote an immune system. And these immune system are what we call the secondary metabolites. And these are the metabolites that we tend to take from the plants to develop into drugs. Now, if we're spraying our plants with pesticides, what we're doing is dampering that plant's ability to develop these metabolites. And so we're just consuming these ingredients, and they're not rich, which is why a lot of persons say you need to eat what you grow these things. And of course, many of us are not in a position to do that. But if you can connect with the right farmers, for example, grapes have something that's wonderful. It's a compound called resveratrol. having
Yes.
tomatoes that have lycopene. So the right
Yes.
balance of these ingredients, of course, is amazing. But we're not having the fruits. And when we do have them, the fruits might not have the metabolites as much as
Yes.
they should. So, you
Because
know,
of the
and then
damage.
we're consuming and inhaling the smoke and we're having a lot of the jerk meats, which we love, but it's showing that the jerk actually is increasing carcinogenic entities in our bodies. And if we don't have the capacity to fight those entities, then we become predisposed to cancers.
Ooh.
Yeah.
Sorrel, another one. I understand sorrel is.
Yes, sorrel is also, I've never looked at it, but I know sorrel is also one of those superfoods.
Okay, thank you, DP. You know, let me just ask you something about this Henrietta Lacks situation. What was said to be amazing about her situation was that the cells kept regenerating. Is that what having a cell line means? Because at the time, it seemed as if that was the first time that people were having a cell that continued to regenerate. Maybe I'm not using that right terms, but you can correct me.
Sure. So we did have cell lines before, but what we had were cell lines from animals like rats. We didn't have a human cell line. So Henrietta Lacks's cells that are called HeLa cells was the first human cell line, and it happened to be a cervical cancer cell line. And it's not with regenerating isn't the correct term, it would be immortal. So it means that her cells just continue to grow, divide, multiply. Regenerating seems like it's something that more of a stem cell, or it's dead and then it comes back. These just continue to grow.
Right. Okay.
And we're able to, for us, we would prefer to have naturally immortal cells, because when the cancer is inside your body, it becomes a cancer, because what it does is it evades the natural growth pattern that the cells should have, because our cells should not be growing indefinitely. They should grow and they should eventually die. But with the cancer, they just keep growing. So they tend to have that immortal effect, in the body. When we take that tumor out and we grow them inside the lab, we hope that they're naturally immortal in the lab, but we have not been able to find naturally immortal lines. So what we do is we induce immortalization. So, we tamper with the biology of the cells to ensure that they are immortal. But what's important for us is that when we do that tampering, we don't change that genetic fingerprint of the cells. And so far, based on the different methods of immortalization that we have seen, or seem to be one of the safest in terms of preserving that genetic fingerprint of the cell lines.
Thank you. And since I don't see any more hands, is there anything else in the chat, President?
No, there is a hand up. Julian Bailey.
Okay, please, I can't see it. Go ahead.
Okay, good afternoon, Dr. Badal. Very interesting presentation.
Thank you,
I just wanted to ask, so for the prostate cancer cell lines, are they available locally for other scientists who are doing similar work?
Not locally, not yet. We don't have that capacity. It is the ACGR PC28 cell line is available to be purchased from a company called ABM.
Okay.
That company is located in Canada. Of course, it would be wonderful if we could be able to supply our local researchers or researchers in the region with the lines. But to do that, you need to have like a master bank set up where you are mass producing, and we don't have that capacity at the time. So for now, we have licensed the line with that company. And when persons approach me, I say to them, for now, ABM has the line that you can purchase.
Okay, well, at least there's an option to be able to get it. My other question is, so a lot of your work has been done on anti-cancer drugs. Are you planning to do any work in carcinogenesis per se, or carcinogens?
So my PhD component, the SIP work, that was looking at compounds that could inhibit the activity of cytochrome P451 enzymes. So you have many cytochrome P450 enzymes, and the one family that has 3 members, 1A1, 1A2, and 1B1, those 3 members have been highly implicated in carcinogenesis by producing highly toxic compounds. That's not a work that I'm doing now, but Professor Rupika Delgado, who is from the Natural Products Institute, I believe that she continues on that path for that research, but that's not my focus right now.
Okay, thank you.
No problem.
And since we're gonna have to wrap, I wanted us to take the privilege of asking the last question.
All right.
Unless,
Before you do the
is there another one?
see that you've asked 2 before. There are 2 more in the chat, so we could probably end with those 2.
Sure, please.
Okay. So the first one is from Karim Smith, and it's on the note of feeding cell lines, soursop, guinean, etc. What are your views on early nutrition intervention, ENI, prior to treatment modalities?
I am not, I can't speak too much on that. I believe that that's more for the clinicians and also for persons in that area. Of course, if I have close family members to me, diet is very important. Exercise is very important. And I know from reading, diet rich in sugars is not good when you're trying to treat cancers because they love the sugars. So starving them of that, I am, from my reading, is one approach. And so that's something that I would recommend to persons who are close to me. But once again, you have to be careful what you say to persons. I'm not a clinician. I'm a biochemist, in the lab, trying to use these models to develop precision therapies later on for persons with the disease. So I can advise, but I'm very cautious in how I relay certain things.
Well, thank you, Doc. And I think that's because the second question was along that line. So I think you've basically answered it with the answer just now. So I think on that note, we can close off our question and answer section. So DP Stewart?
I just wanted to get in this last one.
Okay.
Please. Jack, what does the road look like? You said that developing drugs is a whole different thing and it's a way away. But can you give us an idea of what does the road look like between where you are
and where we need
to go and when we get to the drug stage?
The road looks like what it looks like. I need support. I actually have been trying to meet with different persons from public sector, private sector to get momentum, interest on the work. Because for us to get to where we need to go, we need a state-of-the-art facility that does on mass scale the cell line development and the drug testing, and where we employ all the current models, because importantly in this arena is you have to be on top of what the new models are. So in saying to you, the second stage in the drug development pipeline is animal models. The NIH is coming out saying they want to remove animals. So what is going to replace animal models? 3D models, organoids, organ on a chip. These are all techniques that require specialized facilities that require money. And so the road and the time that it takes us to get there is dependent on the funding that I'm able to secure, because we have the team, we have the drive, the know-how, and if we don't have the know-how, we can connect with persons who have the know-how to get this done. So the limiting factor at this point is the funding. And my dream and my hope is to have a facility that not only focuses on the research, but focuses on the treatment. A facility that any person who is Black or Caribbean here in the region, in Africa, in America, wherever in the world, who looks like us, who develops cancer, they can come here and know that whatever we give them will work for them. And so this is not just about research. This is about translation. This is about policy making. I would love if we could achieve that during my lifetime. And that's what I'm pushing for. They typically say that to develop a anti-cancer drug takes anywhere between 10 to 15 years, but with AI now, we are fast tracking that. And if I can get the money, and if you guys want to help me to raise the funding, I'm certain we can minimize the time, the road and the journey to getting there. But just know that I am here. I have a team of persons who are passionate about this, and we're not giving up. We've come too far, and we also know that we have a long journey ahead. And we are prepared every day we take it. As Stuart knows this, he's been trying to get me for a while, but I have to balance the talks because the work is the priority at this moment. And we're doing that. So it can take a long time, and it can take not so long, depending on the funding that we're able to raise. But that's one of the things that I'm working on in the background.
Thank you. Back to you, Prez.
Thank you, DP Stewart. Thank you again, Doc. And to do the official thank you, I'll invite President Giovanna from the Kiwanis Club of Barbican to do the vote of thanks.
Good evening again, everyone. So on behalf of the Kiwanis Club of Kingston and the Kiwanis Club of Barbican, I would like to extend our sincere thanks and appreciation to you, Dr. Simone Badal, for taking the time to join us this evening for delivering such an insightful and inspiring presentation on No Cell Behind. Your dedication to advancing cancer research and scientific innovation is truly commendable, and we are grateful for the abundance of knowledge that you have shared with us tonight. Again, I would like to extend my appreciation to the Kiwanis Club of Kingston again for partnering with us to host this joint meeting. Your collaboration and support truly reflects the spirit of Kiwanis fellowship and service. I would like to extend special thanks as well to all the members who participated in the various agenda items this evening, which includes those who led the opening formalities, facilitated the program, and supported the smooth running of this meeting. Your contributions are greatly appreciated and helped make this meeting both meaningful and engaging. Finally, but not least, we'd like to thank all the Kiwanians and guests who joined us this evening for your presence and continued commitment to service in our communities. Over to you, Prez.
Thank you.
May I just interject something, President? I'd just like to apologize to our guest that I'm not able to show her certificate of appreciation because of the problems I spoke of earlier before she came on. And I'd just like to add one other thing. The person who did your introduction said you were one of his tutors. He's a doctor, medical. So when I send you the tape, you'll be able to see who it was.
Okay.
So thank you again. Thank you.
Well, you were his tutor, not him being your tutor.
Oh, is that what I said? My apologies.
So it's one of your students, Doc.
Okay.
Okay.
And I was going to say that DP Stewards will find a way to get your certificate to you. We'll make sure that you have it. Thanks
No
again so
problem. Thank you
much.
so much for having me.
You're most welcome, and you're welcome to come back.
Okay. All right. Thank you.
All right,
Thank
Doc.
you again. Bye-bye.
So we'll move to our Sergeant at Arms from Kiwanis Club of Barbican.
Hello, hi, good evening again. As your Sergeant of Arms tonight, my job is to bring a little fun and fellowship to the meeting. And, maybe collect a few friendly finds along the way. At this moment, I invite anyone to just share one word that describes their Easter weekend with us. You can also type it in the chat. My word is busy. Anyone else? Oh, Andrea is relaxing. Orville said blessed. Stephen said restful. Dr. Marlin said quiet. Director Matthew said busy and restful. Joshua said wonderful. Wow, Karen said fun, must be nice. I'm seeing astonishing. I'm seeing short. Yes, amen. Amen on the short. I'm seeing appreciated. I'm seeing fun filled. I can see yours was fun filled. DLG, that one, as you know, we see that you were at the beach in the background earlier.
I went on a boat ride around the harbor on
Okay,
Saturday.
yes, I'm seeing very enlightening, interesting, and informative. Oh, seeing informative, peaceful, and relaxing, exciting. Wow, these are great words to describe, a Easter weekend. Maybe anyone.
My word would be the character games.
Oh, okay. If anyone can share, if you could instantly, right away at this moment, master any skill, what would it be? He says making money. Amen. Amen to that. And just a shape shifting. Anyone else? Singing. Agree with it. Sing and dance. Presenting. Play the cello. Interesting one. That's very interesting. Learning. Sounds good. Anyone else? Okay. Well, at this point in time, we move on to the start itself. So the first one would be show of hands of anyone who attended more than one meeting, Kiwani's meeting for April. I know it's April 7th, but for the persons who have attended more than one meeting so far, it shows, your amazing dedication to Kiwanis. And what better way to show your dedication than contributing to a fine of $100? So each member who have joined more than one meeting, whether board meeting, regular meeting from your club or other clubs, we'll be charging you a fine of $100. As well as for everyone online that has a display picture and who showed their faces on the camera will also be fined $100. First, do you have any special surge on your side?
No, but I'm going to just go very blanket in the spirit of Easter, and we remember, we reflect on what Easter means and that great sacrifice that was made for us. I'm going to ask the members of the Kiwanis Club of Kingston to continue with that sacrificial mode, and each member will be asked to pay $300.
Sounds good to me.
All right.
Billy, you said 3 meetings in I hope you pay a fine.
All right, thank you, Praise. We will
you.
now, is Secretary Dion unknown?
I
Dion,
was saying that the Sarge is very generous, President Zuri. Not allowing for enough money to come in the coffers. $100. Yeah, all right.
Fine him, President.
Too generous.
I don't know why he's getting in President Javana's business. So in that case, DLG, you can pay an addition for some members, for some of our members. So we give you a tax. So on your 300, we'll put in a 100% tax. All right, DLG?
Not hearing.
Oh.
I'm hearing. I heard you. Just joking.
Okay, because you not hearing would have been an additional 50%.
All right, sir.
All right. Is Secretary Dion on now?
No, she had to step out due to work, so I'll be doing our notices and announcements.
Okay.
Okay, so on this Saturday, April 11th, we will be executing the second stage of our Melita Relief Project. We will be heading to Leith Primary and Infant School on the border of St. James and Hanover. For this stage of our project, we will go beyond providing care packages to students and their families, but will also be actively assisting with the physical improvement of the school environment, where we'll be restoring and improving the school's physical infrastructure, creating a safer, more welcoming and functional learning environment for students and the staff. We are currently looking or asking for volunteers to, come down with us to assist with the execution of our various activities that would be taking part in. We're also asking for donations in materials or financial donations to our project. Feel free to reach out to myself or any of our members, or shoot us an email if you'll be interested or if you'd like more information on our project. On Saturday, April 18th, we'll be having our monthly joint project with yours truly, Kiwanis Club of Kingston, where we'll be doing our Mary Atkins Night Shelter feeding on Hanover Street at 9 a.m. And on Sunday, April 26th, we'll be having our quarterly church service and birthday celebration. You guys can look out for more information on that. And that is the notices and announcements from Kiwanis Club of Barbican. Thank you.
All right, thank you, Praise. And at this time, I'm going to say thanks to you all for being here and ask one more thing of you, that you raise your imaginary, literal, figurative glasses with us in a toast to Kiwanis International and to Jamaica Land We Love. This joint meeting of the Kiwanis Club of Kingston and Prez
Barbican
is now terminated. Have a good evening, everyone. Thank you.
Thank you too.
Thanks, guys.
Did anyone mention about the strategic planning meeting training tomorrow? Not sure if I missed
No,
it.
it wasn't. It wasn't. I admitted it. I hadn't noted it.
Yes, tomorrow I think it runs from the 8th to the 9th. So it's 2 days training.
Wednesday and Thursday, 7 o'clock.
Yes, 6:30 to 8:30 each day.
6:30.
You didn't say, is it for the clubs? For the clubs?
Club coaches, mainly.
Facilitators, club coaches, yes. It's intended to train the coaches or others who can impart to the various clubs or, well, who may be coaches for clubs. Yes.
Okay.
It's strategic plan.
Yeah.
Gentlemen, I don't know if the DLG Desmond can, but because of the issues that I was having,
yes,
I am not sure if I'm gonna be able to play the music. So if Desmond, if you can step in, that would be good. But I'm still having some issues that I have to really figure out why I wasn't able to share, what I was trying to do. But I'll figure it out eventually. But I don't think I'll figure it out for this evening. I'm still trying, but I haven't figured it out yet for this evening. But I'm gonna keep trying for now. But Desmond, if you can play some, go ahead.
All right. I will see what I have here.
Where is Stuart? Stuart, I have been kicked off at least 7 times and I still try.
You have been?
The internet is
you have been signal strength.
unstable. Have a great evening,
sorry.
everyone.
Okay,
Thank
okay,
you.
DLG.
Thank you.
Let me just stop this anyway.
Um,
Don't start any music until I tell you. If you
no,
are gonna.
I'm searching.
Yeah, because I want to stop this.
Can you hear me?
I can hear you.
Okay, I'm searching.